All right, so let's get cracking here. All right, now, problem from when we talked about this last time, and this is just a general observation, is the book does not really go that deep into what we're talking about. And that can be a problem. Okay, so for example, if I want to tell you how to get to Atlanta, right, I might say, okay, go out here and turn to the left and go past Starbucks and turn to the right and go around in a circle and then keep on going past the Ferris wheel and the roller coaster and you'll be in Atlanta. That works, right? That's an accurate description going down I-20. But it's not a very satisfying answer. Well, not to me at least. Conversely, I could give you some massive detailed list here, can I? But is that beneficial to you? Not really. The problem here is this is not enough information, and this is way too much information. Okay, so I'm kind of faced with a conundrum here. We want to give enough information that is meaningful and understandable, but not give you so much information that it's now a biochemistry class. Any biology majors? Chemistry majors? No? Well, we have a class in it's a chemistry class here called biochemistry, and it really, you have to learn every single step of the whole process. So we're not trying to get that level of detail. So I want to kind of give the question we're dealing with. The question we have dealt with here is how do we get from this? How do we get from hamburgers and pixie sticks to running? Right? How do we get there? Right? Or how do we get from our pixie sticks and our hamburgers, or our pork sandwiches, or whatever we had today, to something like our heart beating? Right? There is a connection there. So another way we're trying to, this is how a muscle contracts. This is a general description of how a muscle contracts. And this requires ATP to function. Okay, so this requires, it might be hard to see, but here we have an ATP here working. So to have a muscle contract, we need ATP. Okay. So in a word, what are we talking about? We're talking about something called metabolism. That's the basic thing of what our next test is on. So basically, here's the big picture of what we're doing. Okay, first we start over here with that hamburger and those pixie sticks, and those sound really good right now. We then break those down into our macromolecules, our carbohydrates, our triglycerides, or our lipids here, or here is our protein. We then break that down further into ATP. This is our ATP molecule. And then we use ATP for things like muscle contraction and actually muscle relaxation as well. So that's the basic point of what we're doing. We're going to be particularly concerned with this arrow right here. This arrow basically is the whole chapter. How do we go from these molecules to ATP? That's the point. Okay. So first we break them down. This is a digestion process. You cover this in the second semester of introductory biology. This is digestion. This is what we are concerned about. We are converting things like carbohydrates, lipids, proteins into this energy carrier that we have come to all know and love, called what? What's it called? ATP. ATP. All right. Okay. So let me, and I'm going to say this a few times because this is the whole point here. The process that transfers energy of the macromolecules into ATP is the point of this chapter. Okay? Say it again. The process with fireworks, hooray, the process that converts the energy in these macromolecules. These macromolecules have energy. Protein contains energy. Lipids contain energy. Carbohydrates contain energy. What we're doing is we're taking that form of energy and making it into another form of energy. We're not making energy, we're converting energy from one form to another form. Okay. All right. Okay. I think I went a little overboard with the effects. All right, but why? Why are we doing this? Why are you sitting here on a Friday listening to me blather on about this? We just kind of said it. Why are we doing this conversion? So we can have a form of energy that different processes in our body can use. Okay, we're using pumps to move ions for the nervous system, for our neurons to work, for our brain to work. You know, we're moving myosin heads in our muscles to cause a muscle contraction. We're turning on pumps to, for example, to have your muscle relax. So we need this energy to do this. But we cannot get that from glucose as is. We have to do a series of metabolic reactions to bring it to that ATP level so we can use it. Okay. Given that analogy that I just so am in love with, apparently, I'm probably going to marry this analogy at some point in my life, is we start with our macromolecules here. Macromolecules are packed full of energy like a $100 bill. But can I use a $100 bill in a vending machine? No, I can't. I can't do that. Okay? So what I got to do is I need to take these and make many of these. Okay? I need to take my $100 bill and make many $1 bills. And those $1 bills I can use in a vending machine. Conversely, or in the same idea, I can take these ATPs and use them for things like muscle contraction, muscle relaxation, other processes in the body. That's the whole point of this operation. That's why we're doing this. Okay, that little arrow right there, that is our focus. That's what we are concerned about. Okay? That little arrow right there. We go from the macromolecules here to the ATP form here. Macromolecules have energy, ATP has energy. We can't use the macromolecules over here for these processes. We have to get them into a particular format we can use them. And that's why we do all this. Okay. Consider that mule, horse, beaten, dead. How's that? Okay. Or say another way, we're just trying to get energy into an ATP form, which is a usable form. That's the whole point of this. All right? Good so far. Outstanding. All right. Now, before we do this, we've got to introduce this idea of oxidation and reduction. Oxidation and reduction. So an oxidation is when an electron is lost. A reduction is when an electron is gained. And I'm going to give you a great mnemonic here. What do you call those big things you get oil out of from the ocean? An oil what? An oil rig. Have you heard this mnemonic? Yes? No? Maybe? Oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. So if something gains an electron, it is what? Reduced or oxidized? If something gains an electron, it is reduced. If something loses an electron, it is oxidized. And you might say, whoa, 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 how can you be reduced and gain something? That's crazy talk. Well, what charges an electron? Negative. So electrons are negative. And what would happen to the charge if you add a negative? It goes down. Its charge is reduced. So reduction reduces the overall charge. That's the idea. Really, all these namings happened before we understood everything that was happening on a subatomic level. Okay. But if you remember this mnemonic, oxidation is a loss, reduction is a gain, you'll be fine. This is an important concept when we start talking about metabolism. Okay. All right. Questions at this point? We're good? All right. So, we're going to need to move things like protons. We're going to need to move electrons. And I kind of used this analogy last time, and I'm going to come back to it several times today. If you need to move beer from one area to another area, so let's say you're at an ABC store. I got beer here at this ABC store, and I'm going to have a party all the way in Heflin. So I don't know. Do I just say, okay, turn the tap on and hope that the beer winds its way to Heflin, 40 or 50 miles away? No, I don't. What do? You buy a keg, yes. You buy a very large container to put said alcoholic beverage in, assuming you're above the age that you need to be. Well, we're going to do the same thing here in the cycle. We're going to have kegs for electrons and protons. Okay, because one time we need them, we're going to pull them off one area, and we're going to take them into an entirely different area and use them. So for example, in glycolysis, we're going to pull off electrons and protons, and we're going to carry that all the way into the mitochondria to use it. Okay. Now, here are some of the kegs we're going to talk about. We've already talked about some of these. NAD. Okay, NAD, and we'll talk about that today. That's an empty keg. When we add some electrons and hydrogens, we've got NADH. One we talked about with photosynthesis was NADP. Remember that one? When we filled it full of stuff, we made NADPH. FAD, another empty keg. We're going to add some hydrogens, and we're going to make FADH2. Okay, so you're going to see those multiple times today. Point is, when we make those, we're carrying electrons and protons from one area to another area for a function. Okay. Now, kind of review. When we do this, what do we call this? When we add electrons to that, what have we done to it? We have oxidized or reduced? Reduced. When we remove those electrons and we empty that keg, we have done what to it? We have oxidized it. That's an oxidation reduction. Okay. All right, so let's talk about some of these metabolic pathways. What is a metabolic pathway? Metabolic pathways are multiple reactions stacked on top of each other. Series of reactions, one on top of each other. That's a metabolic pathway. Now, metabolism is broken down into two different parts. We have catabolic reactions and we have anabolic reactions. Okay, say you got a 1973 Nova and you want to make money off of eBay and sell the parts of this 1973 Nova. What are you going to do to it? You're going to blank it for parts. You're going to do what with it for parts? You're going to cannibalize it for parts, right? You heard the term cannibalize? You're going to cannibalize it for parts. You're going to take things off of it. You're going to break it down. Okay, catabolism, are we good on the slide? Okay. Catabolism is when we capture energy for use by the breakdown of something. All right, the other side of this is anabolism, or anabolic pathways. If you're on, what's the juice? If you think of juice, what's the juice? It's what steroids? You can tell I'm probably on it right now, right? Anabolic. Yeah, anabolic steroids. Anabolic steroids, you're building up muscles, right? Okay, anabolism or anabolic reactions are actions that build up things. We're going to use energy and we're going to build up. We're going to make more complex structures. Metabolism is an umbrella term for both of these pathways. All right. Okay, so far so good. All right. So now we're going to talk about glycolysis. And before we do this, let me just give you the skinny on the whole idea here. This is glycolysis relative to the other processes we're going to talk about. Do you have to know everything here? No. No, you don't. Let's talk about some things you do have to know. We're going to start with glucose. And the process we first metabolize glucose is glycolysis. What does glycolysis mean? We said it last time. Remember hydrolysis? With hydrolysis, we split water, right? With glycolysis, guess what we're going to split? glucose. We're going to take a glucose and split that glucose into 2 3-carbon compounds. In the process, we're going to fill up those kegs. We're going to make something called NADH in that process. Next, we're going to then take those pyruvates or pyruvic acids, and we can either go into what we call oxidative metabolism or respiration, or we can continue on to fermentation. We'll talk about fermentation next time. It depends on how much oxygen you have. It depends on cellular processes. We'll talk about why you do fermentation later on. Okay, but there's some choices that the cell will make. Once we have that 3-carbon compound, we're going to go into what I call the Kreb cycle. We've got to prepare it by making it into acetyl-CoA. We'll get to that. The Kreb cycle is going to make a lot of these carriers, FADH and NADH, and that will go on to the electron transport chain, and this will make lots and lots and lots of ATP. Now we're going to talk about each one of these as a separate section. Okay, we'll get to each one as we go along, but this is the overall schematic of what we're talking about. So again, we're talking about glycolysis. We're just going to talk about what happens in glycolysis. Now, these diagrams are a little bit more involved than what's in your book. You don't have to memorize every single thing going on here, but I wanted to have these so you can have a better idea of what's happening. Instead of it being a bunch of gold spheres here and there, I think this does a little bit better job of conveying what's happening. Not too in-depth, but not enough information that I think is more meaningful. So, we bring glucose into the cell. We bring it in, and we are going to invest 2 ATPs. So, do you see ATP has 3 phosphates, right? The T is tri, right? All right. So here, this 3rd phosphate is going to be added to our glucose. Next, this 3rd phosphate will be added to the glucose here. So now we've added how many phosphates to our glucose? 1, 2. We've added 2 phosphates to our glucose. And do you remember we talked about the structure of phosphate? What did it have? It had phosphorus and what else? Oxygen, right? It had that phosphorus-oxygen structure. Those are very negative. These are repulsing each other. Okay? Think about it like magnets. Think about 2 negative charges on a magnet. If I have 2 negative charges close by on a magnet, are they going to repulse or attract? Repulse, exactly. So same idea here. These are going to repulse, and what they're going to do is they're going to make this bond right here not so stable, because those things are repulsing, and we'll have an enzyme that will come in, and when it grabs onto this, it can more easily break this center bond because it has these 2 charges on it. So when it does this, guess what it's going to break it into? How many pieces? 2. It's going to break our 6-carbon compound into 2 separate pieces. Okay. How many ATP did we have to invest to do that? 2. Okay. We had to invest 2 ATP to do this. So what do I want you to know about this diagram? 1, that when glucose comes into the cell, we're going to add 2 phosphates to it. This will cost us 2 ATP. But this helps us break that 1 compound into 2 separate compounds. Makes us break that 1 6-carbon compound into 2 3-carbon compounds. Right? We have 6 carbons. We have 2 3-carbon compounds. Okay. Now, did this require oxygen? You see any oxygen factoring in there? No. No oxygen was required for this. We're not going to see oxygen use in glycolysis. Okay, no oxygen was required. So now we have broken that glucose, and hence the term glycolysis, right? We're splitting glucose into 2 3-carbon compounds. All right? Next, what are we going to do? We're going to make, well, let's say this. We're going to fill up our keg. So here we have NAD. Here we have NAD. And again, there's two separate pathways happening here. Okay? Because we've split that carbon into two, right? Or that 6 carbon into 2 3 carbons. All right? So now we have our NAD, and NAD will gain a phosphate, a hydrogen and electron. And here the NAD will fill up with an electron and a proton, a hydrogen. You see, it's taking something from, ultimately, what the box in green, this and this ultimately comes from what? What's in green? This and this. What did those come from? The split product ultimately coming from? You're right, glucose, correct. So if these backtracked, right, this would be glucose. So what's happening here is NAD is taking some high energy things from that byproduct of glucose. Okay? It's filling the keg up, so to speak. Now when it transfers an electron, it's getting an electron, what's it being done? Is it being oxidized or reduced? Reduced, exactly. So we have a reduction here and a reduction here. So what I want you to know about this, I want you to know that the next step of glycolysis after we split it is we're going to make 2 NADHs. 1 NADH per 3 carbon compound. Okay. That's it. Now we've got to do a payday here. We've got to make some money. We've spent a lot of ATP, right? We've spent 2 ATP. But we need to make some now. So what we're going to do is we are going to make ATP from ADP. So do you see we have a blue phosphate here? We see that? And below it we have an A and 2 Ps. That's ADP. That blue phosphate, I'm just drawing it as blue, will add here. So we've added a third phosphate and made, guess what? Guess what we made? ATP. So we've made a single ATP. What have we lost in the process on that 3 carbon compound? A phosphate, right? See how we have 2 phosphates on it before, and now we have how many? 1. See how we have a P and a P on both sides, and then the middle one we have a P on one side and it's missing it on the other side? Okay, we've made an ATP. We continue on. Another phosphate comes off, and let me redo that. Here I'm going to draw that phosphate as purple, and now it goes to make another what? What have we made? ATP. So ATP number 2. A similar thing is going to happen with the other 3 carbon compound. Here, this phosphate comes off and is here. This is our 3rd ATP. This phosphate comes off, and now comes to this ATP. So we've made 4 ATP. All right, so let's go over some numbers here. How many ATP have we made as a gross number? 4. How many did we have to invest to get there? 2. So if we made $4, we had to spend $2 to get there. How many have we made net? 2. We've made 2 ATP net. But what else have we made? What's at the bottom? We have this pyruvate or pyruvic acid. Don't worry about the difference with pyruvate and pyruvic acid. In the chemistry jargon, we call them a conjugate acid-base pair. Difference is academic for the purpose of this. We're going to treat pyruvate the same as pyruvic acid. So if you see a different term, it's not a difference that is important at this point. All right. So the end product of this glycolysis pathway is 2 pyruvates, as well as 2 ATPs. And how many NADHs? How many NADHs did we make? 2. We made 2. Remember, initially, each of those made right here. This one right here, this is 1 NADH being made. This is the 2nd NADH being made. All right? Okay. All right, so what's the key points here? One, we made a very small amount of ATP, right? We made net 2 ATP. We had to use some ATP also, right? So we used some ATP. And we made 2 pyruvates also. We made 2 pyruvates. Now these pyruvates, we can continue on to another reaction and get more energy out of them if we can. But for that, we need oxygen. So we'll get to that later. So what are some advantages to this? First of all, no oxygen. You go back and look at those diagrams, you're not going to see a single place where oxygen comes into play. It's not there. We don't need oxygen for that. 2, it's relatively fast. It's happening in the cytoplasm, in eukaryotic cells, really happening at a very fast pace. So it's not making a lot of ATP for each reaction, but it can happen fairly fast. Disadvantages, small amount of ATP. We'll talk about the Kreb cycle electron transport. That's going to make ATP 36, 38 ATPs. It's going to make a lot of ATPs. This makes 2. Okay. All right. Now the real magic happens with what the cell does to the pyruvate. Okay. Because there's multiple things it can do with pyruvate. And that's the next topic we're talking about. Okay. So now let's talk about the processing of that pyruvate. So we're going to start looking at the Kreb cycle, or aerobic respiration. Now before we can take pyruvate, pyruvate still has a lot of energy in it. It has a lot of chemical potential energy in it. But we got to prepare it for the next step. We want to take that compound. How many carbons is in pyruvate? 3, right? 3. We got 3 carbons here. And we want to take it into something called the Kreb cycle. We'll get to that in a second. But we can't, it doesn't fit in there because it has to have 2 carbons. So we've got to make something so we're here, and we have 3 carbons here. We want to make it into something that has 2 carbons so we can enter into the Kreb cycle. And that's the formation of something called Acetyl-CoA, or Acetyl-CoA. So before pyruvate can go into the Kreb cycle, this is pyruvate right here. Pyruvate has to be processed from a 3-carbon compound to a 2-carbon compound. Now something very important biologically happens here. We've got to do, if we have 3 and we need 2 carbons, what do we got to pull off? A carbon, right? This has 3 up here. This has 2. We've got to get rid of a carbon. What do we breathe out? Carbon dioxide. That and bad breath. We've had onions today. But aside from that, carbon dioxide. All right? Well, we're going to get one of those carbon dioxides. Some of the carbon dioxide, actually a good proportion of that carbon dioxide you breathe out is from this process right here. You're preparing that pyruvate to enter the Kreb cycle. And to do that preparation, you've got to pull off a carbon dioxide. Okay? That's one of the major reasons we breathe out carbon dioxide. Okay. Now, do you have to know this entire process here? No. No. Just see the concept of the operation. We start with a 3-carbon compound. We pull off a carbon dioxide that yields a 2-carbon compound. We make an NAD to NADH. Again, that is for that keg idea. We're filling up the keg. And now we are ready to go to what? The Kreb cycle. We can now go to the Kreb cycle to make some more energy. But we have to do this before we go in the Kreb cycle. It's like you ever see those no admittance signs? Okay, no one allowed. Well, this one is pyruvate is not allowed in the Kreb cycle. It has to have a carbon removed to make acetyl-CoA so it can enter the Kreb cycle. That's the point. Okay. So now we are now going to the Kreb cycle. Okay. Let me show you the cycle here and make a point. Do you have to memorize this entire cycle? No. No, you don't. But I want to make a few points about it that are kind of important. First of all, it is a cyclic reaction, right? It has, we're going to see there's a cyclic reaction. We're going to see that at multiple points, we're going to make NADH. We're going to fill the keg up. And here's another NADH. And here's an FADH2. And here's another NADH. We're filling the kegs up with high energy electrons and protons for the next step after this. But we're also doing something else. We are removing carbon dioxide as well. Okay? We have to remove carbon dioxide. This is the other reason why you breathe out carbon dioxide. The Kreb cycle, as it processes, and as it happens in the mitochondria, this will pull out carbon dioxide in the process. That's the other reason we breathe out CO2. Okay. We're also going to make a little bit of ATP in its form of called GTP, but for now we'll just say it's equivalent to an ATP. So we're not making a lot of energy ATP-wise in the Kreb cycle, but we're making the carriers necessary to make that energy later on. We're filling the kegs up. We're not making a lot of ATP, but what comes of this, and this will make a lot of ATP later on. Now, for one glucose, how many Kreb cycle reactions do we see? How many times will this happen for one glucose? Good question. How many times? Theoretically, what's the maximum time pyruvate can go from here and do this? Once or twice? Well, what? Well, okay, well, what goes in this? Pyruvate, right? How many pyruvates per glucose? 2. So we have 2 turns per one glucose. So these numbers, when you start looking at them, you got to put a 2 on them when you start calculating ATP amounts. Okay. So just some highlights on this. A lot of this is just for your own personal reading. But the first step, Acetyl-CoA, which was made from what? Pyruvate, right? Pyruvate made acetyl-CoA. In the process, we removed a what? Carbon dioxide, CO2. Okay, we then enter acetyl-CoA into the Kreb cycle. Okay. Acetyl-CoA has 2 carbons, and it combines with a 4-carbon intermediate to make a 6-carbon compound called citric acid. Now, you don't need to know all of those names, but I do want you to see that we're adding a 2-carbon compound to an existing 4-carbon compound to make a 6-carbon compound. Okay. All right. So now we have 6 carbons right here, right? Let's just count the carbons and make the major points. We've got 6 carbons here, right? Okay. Now, we're going to pull off some of those electrons and protons for our keg right here. Is this a reduction or an oxidation? Is NAD being reduced or oxidized? Not oxidation. It's gaining electrons. If it's gaining electrons here, it's being what? Reduced. Reduction is gain of electrons. So it's being reduced. Now, yet how you said it though, technically something is being oxidized. Because when you see a reduction, you typically see an oxidation. So our compound here is being oxidized, right? That's what you're probably, okay. So you're correct. This is being oxidized, but then the NAD is being reduced. And that's why I call it redox reactions. They kind of go hand in hand. Right? Something gains, something's going to lose. All right. Now, we also bring off a what? What's that? A CO2, a carbon dioxide. Again, why we're breathing out carbon dioxides. Right? next we're going to make, essentially we're going to call it an ATP. Okay, technically it's not an ATP, but for purposes of this, we're making an ATP. That's all we're making, just making an ATP. Right? Then we're going to make something called FADH2. Again, another electron carrier, another keg is being made. And then another NADH is being made. All right? Okay. So what's the whole consequence of this? We have made a lot of carriers, right? We've made a lot of energy-containing carriers. We've made, let me write all this down here. What have we made in all this? Let me turn the light on here. What have we made in the Kreb cycle? Let's kind of go back and do a summary. What entered the Kreb cycle? Pyruvate to what? Pyruvate formed what at the start of this? The Acetyl-CoA, right? Acetyl-CoA, either way we want to say it. And this has how many carbons? 2. And when we did this reaction, pyruvate has 3 carbons. When we did this reaction, what came off? CO2. And also that NADH. That full keg, if you will. All right? That acetyl-CoA enters the Kreb cycle, right? That enters the Kreb cycle. Then what's next? We go in a circle around the Kreb cycle, and what do? We release, in a stepwise reaction, we start releasing what? NADH, right? And when we release this, what else do we release? CO what? 2. CO2 is released. Yeah. And we'll keep on going around, and we'll release NADH, we'll release FADH2, we'll release some ATP, right? And we've got the numbers on the thing, and we got CO2 and so we're releasing multiple energy carriers in this reaction. Okay? So we're releasing some energy carriers, we're releasing some carbon dioxide. Okay? Now, we're not making that much ATP from this per se. But we're making some carriers for that ATP. All right? Now, when we go through this reaction, when we release 2 carbons, how many carbons started here at the very start? 6, right? How many started this from acetyl-CoA? 2. So if we have 6 here and remove a carbon dioxide here, how many do we have now? 5. If we remove another carbon dioxide, how many carbons do we have now? 4. We go back, and that's why you're removing those carbons, because now what combines with your acetyl-CoA has 4 carbons. Acetyl-CoA has 2 carbons, and then it keeps that carbon number constant. Okay. All right. So that's the important thing. So you have to know every single intermediate? No. No, not at all. You have to get the idea that we're making energy carriers, the NADHs, the FADH2s. You have to get the idea that we are removing carbon dioxide from the reaction. That's why we breathe out carbon dioxide. You're making a little bit of ATP, but not a heck of a lot from the Kreb cycle. All right. Now here comes the big payday. All right, here comes the wind-up pitch. We're going to start making a lot of ATP. And that's the third part of this reaction. Remember this picture we had? I said we'd take it to the electron transport chain. Kind of remember this? All right, we're going there. So what we're going to do, in this membrane here, this is different for prokaryotic and eukaryotic organisms. All right? We're going to talk about eukaryotic organisms. We're going to talk about this in the mitochondria. What we're going to do is we're going to take those electrons that we carried in the NADH, that full keg. Right here, we got a full keg right here. We're going to carry those, and we're going to carry them to the membrane of the mitochondria. And we're going to use the energy that those contain, because they're energetic, we're going to use the energy that those contain to drive the formation of a proton gradient. We're going to make a lot of protons in one area, and what's going to provide those for us is the NADH and the FADH2. Okay. So here we go. Here is our keg right here. NADH, right? We have NADH. NADH provides the electron through here, and it will also do something else. It will move the protons, here shown in blue, to the other side. See what's happening? Here's our keg, NADH. It will provide energy to move a proton to the other side, and it will also do what? Provide electrons. Okay. These electrons will then move through something we call the electron transport chain. As these electrons move through the electron transport chain, we're going to move other protons to the other side. We're going to make a concentration gradient of protons. Okay. Here, if you see the mitochondria, we are doing this between the two membranes of the mitochondria. Okay. What do we call the mitochondria? We called it what? The what house of the cell? The powerhouse of the cell. This is where this is happening. Okay. We're going to go through this stepwise, but I'm giving you the overall view right now. We're then going to take these protons and we're going to move them through ATP synthase. Remember we talked about ATP synthase, that thing that's spinning around, all that? Same thing here. We're going to move those through ATP synthase, and we're going to make ATP as a consequence. As these move through the synthase, it will physically spin, and it will provide the energy to make ATP. The big payday. Typically, we're going to expect to get about 3 ATP for one NADH and 2 ATP for an FADH2. Okay, that's an approximate payday from those. All right, now, we got a problem. What's the problem? Do we have a cosmic rug that we can, because we're moving electrons through here, aren't we? Yeah? What would we've got two problems here, really two major problems. One, we're moving electrons, right? Something has to take all those electrons, right? We don't have a cosmic rug that we can sweep electrons into, do we? Let's go back and talk about concentration gradients. What happens if we have two concentrations of equal amount? Do things move from each side? Well, if they do, it's going to balance out, right? So we've got to have a high concentration on one side and a low concentration on the other side, right? But if we're always moving protons through here, what's going to happen to the concentration of protons on this inner side part? It's going to start going what? It's going to start going up, exactly. So we've got a double whammy problem here. We're getting a lot of protons here, problem number one. Problem number two is we've got a buildup of electrons. It's kind of a problem, isn't it? Why do we deal with it? Well, what do we breathe in? Oxygen. Oxygen is what we call a terminal electron acceptor. Oxygen will combine with the electrons and some protons to make, guess what? H2O, water. It's going to make water. Now let's go back to photosynthesis. What did the plants use for its source of electrons and protons? Well, not light was the energy, but then it took that energy and it broke something apart for the hydrogens and protons. Water. And remember, we took that water in photosynthesis and we broke it apart? And what was released as a byproduct? What did plants breathe out? Oxygen, exactly. So kind of at the point here. So plants, this little number right here, they had water. They split into the protons, right, and the oxygen, right? Oxygen coming off as a byproduct. We take that oxygen, and guess what we do to it? We add what? H plus of the protons and some electrons. And guess what we make? What are we going to make? Take this oxygen in us and add some protons and electrons, and guess what we make? Water. Okay. We call oxygen a terminal electron acceptor. Okay. Oxygen is a terminal electron acceptor. And again, it's maintaining that concentration gradient, right? Because we have a lot of protons going there. If there's no gradient, the whole thing breaks down. If there's a buildup of electrons, again, the whole thing breaks down. We've got to get those protons cleared out to a degree. We've got to get those electrons cleared out. We dump them into oxygen. Oxygen then becomes water. The hat trick. All right. And then don't forget this relationship here. Plants are going to use water, source of electrons and protons, release oxygen as a byproduct. We're going to use oxygen, add protons and electrons, and release water as a byproduct. It's a nice, beautiful cyclic reaction there. Now, we saw in this diagram what was carrying the energy to these. What was carrying the energy here? Most of it. What compounds? What carried those protons, those electrons to this transport chain? What two compounds? No, but what kegs? We call them as kegs. What brought them there? NADH and what else? FADH2. All right. So what did that NADH? So we kind of saw that NADH and FADH2 were critical importance for making ATP here, weren't they? Where did those come from? Where? Glycolysis and where else? The Kreb cycle. Kreb cycle and glycolysis will make a lot of those carriers. They will fill the kegs up. Glycolysis is happening in the cytoplasm. We've got to carry that to the mitochondria. That's why we fill them up, right? You got beer in Heflin, right? Or no, beer here, and you want to get that beer to Heflin. Are you going to open the tap up and hope that it winds its way to Heflin miles away? No. You're going to put it in a keg, and you're going to carry it there. The same thing happens here. We want to get something from point A to point B. We just can't let it get on the ground and hope that it finds its own way there. We've got to put it in a container and carry it to the source where we can use it. That's what NADH and FADH2 do. We take it from one area and we carry it to the electron transport chain in the mitochondria so we can use it to make what? Make what? What is it going to ultimately make here? ATP. Exactly. An energy currency of the cell that we can use. Okay. So two important questions here. Where do the electrons and protons come from? NAD, FADH2. Where did we make those? Kreb cycle, glycolysis. What did we get the raw material from? In this example, glucose. Now I've put several things in here, and it's just to give you an idea of what's happening. But it's really important to see some major points here. Again, NADH, FADH2 provide some raw materials and the energy. They go through this transport chain, and we build up a gradient in the mitochondria between the two membranes of the mitochondria. We then allow the protons to flow through ATP synthase. As they flow through ATP synthase, we make what? ATP. But we have a buildup of electrons and protons. Where do we dump those to? Oxygen. The oxygen will take those protons and those electrons and make what as a result? What? Well, water will be the result. Oxygen and hydrogen will combine to make H2O, water. That's why we have to bring in oxygen. Because if this does not happen, tell you what, let me turn your oxygen off for a few minutes. Can I do that? No. You see why you need oxygen, right? If you don't have oxygen, this whole system falls down like a Jenga block building, literally. The whole thing comes crashing down. Okay, it's that important. That's why you need oxygen. Okay, terminal electron acceptor. Okay. All right. Now, I've done it multiple times here because I know this is a complex subject. So I want you to look at this and kind of get an idea of what's happening. But I don't want you to memorize every single slide if we haven't talked about it. Because I know this is a conceptually hard topic to get. I get that. Okay. All right. Okay. Now let me just show you some of the numbers from this, and I think you're going to see why we do this. Now we talked about glycolysis. Glycolysis gave us how many net ATPs from glycolysis? Net. 2, right? We spent 2, we made 4. Great, we made 2. Let's look at some of the numbers from that Kreb cycle and oxidative metabolism. I'm seeing some pretty big numbers here. 34, 40, 38. The number is actually going to vary between bacteria and eukaryotic organisms because of movement across the mitochondria is going to take some energy. That's a little bit more in-depth than you go into, but I see a big difference. Which is more, 36, 38 over here, or 2? Kaplunk, yeah. It's a lot more ATP. It's a lot more ATP, and that's kind of the point to it. All right, but the other downside, what is the downside to that process, though? It needs oxygen, right? And what's the other downside? Time. That whole process, that seemed really, it has to go here, has to go here, has to go to the mitochondria. It's not as fast as glycolysis. And time-wise, it's not as fast. All right, we'll finish this up with fermentation next time. And then we'll also talk about, and this is a thing that your book doesn't go into, but I will talk about it a little bit. We've just talked about glucose, right? But food's a little bit, we don't eat just pixie sticks, do we? No. We eat protein. We eat what else? Fats, right? We're going to talk a little bit about how fats work. It's not that much more. That whole process, we're going to see how fats actually feed into glycolysis. So we'll talk about how, why we break down fats a little bit also. Okay, we're not going to get super in depth in it, but we're going to talk about why fats also make energy. But it fits into the same framework. All right, guys. Have a good weekend, and I will see you here Monday.